Hello. Okay. There are many factors influencing the effectiveness of various microbial control agents, including temperature. And recall that temperature is the measure of molecular motion within a system or an object. So if you increase molecular motion or decrease molecular motion, this will have a direct effect on how well the antimicrobial chemical will work. Time of exposure or application of a given disinfectant or antiseptic will also have an effect. There has to be an appropriate amount of the chemical in terms of its time used. And speaking of amounts, concentration of the chemical matters greatly as well. One common example of this would be how concentration can be influenced by evaporation characteristics, such as in alcohol. Surprisingly, alcohol has been found to be more effective at 70% concentration when compared to 90%. And initially, you might think that 90% would be more effective because there's a greater amount of the active ingredient. However, 90% alcohol is going to evaporate more quickly from the surface that the person is trying to use to treat the area, and so its antimicrobial effectiveness is reduced. At 70%, there seems to be a magical balance between the amount of water that keeps the active ingredient on the surface and in contact, killing bacteria or viruses. And below 70%, you have a reduced amount of active ingredient, and so it's not as effective as 70. Beyond 70, you get too much evaporation and not enough water to keep the active ingredient in contact with the surface. Lastly, we can talk about the presence of organic matter as a factor. And the term, don't let the term organic matter confuse you. Organic matter is referring to objects in the environment that you're trying to treat, such as skin or clothing. And these types of organic matter can be absorptive. So if they absorb the active ingredient, then the active ingredient may not stick around on the surface that you're trying to treat, and so there can be a reduced effectiveness. A couple of common chemical antimicrobial control measures include peroxygens and ethylene oxide. Peroxygens include ozone. Ozone is represented by the molecular formula O3, and when compared to ordinary oxygen that we breathe, like O2, the extra oxygen molecule in O3, that atom can break off and react and oxidize microorganisms. And similarly, peroxide, otherwise known as hydrogen peroxide, can also act as a molecule that contains an extra oxygen that's unstable. So water has the molecular formula H2O, but then H2O2, which peroxide contains, will be unstable enough for the oxygen to detach and oxidize organisms. And so we can label this as a cidal category of chemical treatments that are used to decontaminate water, whether it's in a hot tub or it's for treatment of water that'll eventually be drinkable, like at the water treatment plant. And we can say that these measures are not sterilization level measures. For the most part, they would be thought of as not relied upon for sterilization, but still extremely effective at decontaminating, at killing most of the microorganisms that they come into contact with. And I'm going to say here, this is that sterilization is not achieved under normal concentrations. But normal would refer to, say, the concentrations that you would see of peroxide at the pharmacy. So if you were to go into the pharmacy and buy some peroxide, it would be at about 3% H2O2. But there are some industrial applications where peroxide could be brought up to a concentration of 50% or greater, and in those instances sterilization can be achieved. But that's extremely uncommon, and at 50% you could start getting into a category of there being explosive amounts of peroxide. And so we'll just stick to the idea that for the most part it is not a sterilant. Ethylene oxide, on the other hand, down below here on the second part of this slide, acts as a sterilant. It is cidal, but it can be relied upon as an agent that will kill all forms of life and at a low temperature. So it doesn't act as an oxidizing agent. It acts as what's called an alkylating agent. And alkylating agents are chemicals like this gas that attach to and act as corrosive chemicals that will destroy various cellular parts of microorganisms. And additionally, I'll add something else on here that's very similar in how it works. And it's not ethylene oxide, but it's also a gas-like application called vacuum gas plasma sterilization. And plasma is technically not a gas in its traditional form. So when you learn about the three basic states of matter, you have solids, liquids, and gases. And then plasma is a special fourth state of matter in which matter, atoms, molecules that are in a plasma will separate their nuclei from their normal association with their electrons. And the electrons can travel around in unusual ways that represent a cidal measure for life forms. And it can achieve sterilization when the plasma comes into contact with the surfaces that you're trying to sterilize. Now, the plasma that's used is typically like a liquid CO2 that is sort of in between a gas and a liquid state. But many different molecules or atoms can enter into a plasma state. And for the industry of sterilization, they typically will use CO2. In this figure, we see a ranking of all the different infectious agents and their relative resistance to various antimicrobial measures. And at the very top of this list, we have prions. And prions aren't alive at all. They are not really a life form, but can be described as infectious proteins. And it's surprising that a protein can be an infectious agent. But these infectious proteins are extremely dangerous in that they can cause diseases such as mad cow disease, and then also other what are called prion-based diseases. And we'll learn more about those during the virology chapter, as that's where they're discussed in the book. Prions are so difficult to neutralize that even ordinary conditions of sterilization will not do the job to neutralize them. So the autoclave has to be changed from 121 degrees Celsius up to about 145 degrees Celsius with increased pressure and time in order for prions to be denatured sufficiently so that they're not infectious. Next on the list, we have endospores. And we've learned a fair amount about endospores in this course so far. And what makes endospores so durable in the bacteria that produce them are the keratin-like proteins that protect the endospore from being degraded by most disinfectants and antiseptics. Those types of chemicals will not penetrate through the keratin, but an autoclave and certain special applications that are labeled as sterilization techniques will work. And the same can be said for mycobacteria. Mycobacteria are also extremely difficult, resistant types of bacteria, and they are protected by a sticky, waxy substance that we learned about called mycolic acid, and that's in their specialized cell walls. And next on the list, protozoans have two different forms. The cyst form is built for survival, and we have understood that they have a cyst wall when we were learning about protozoans. And a cyst wall is designed to resist environmental dangers, including ingestion by the animal host or the human host, where stomach acid is very dangerous, and yet the cyst wall can resist this hydrochloric acid, at least for a brief period of time, as they're then ingested. And once they survive that ingestion and become vegetative protozoa, they still have, in their trophozoite form, a pellicle structure. And this pellicle is another word for a skin, but it's a fairly tough skin, and that makes them somewhat resistant to various antiseptics and disinfectants. And below this on the list we have gram-negative bacteria, and we know that gram-negative bacteria have some elevated resistance because of the presence of their outer membrane structures that help to protect them more than ordinary bacteria that are gram-positive that lack an outer membrane. And then we have fungi that are next on the list, and we know that fungi are fairly durable in that their cell wall has chitin. So the chitin can protect them from, that makes them a little bit better at resisting drying out, and also to osmotic pressure. And next we have viruses. Viruses, particularly without envelopes. Viruses without envelopes simply have a capsid. And that protein capsid shell is something that allows them to survive some exposure to certain disinfectants and antiseptics. And I'll skip over gram-positive for a moment and compare the naked viruses, the ones without envelopes, to the enveloped viruses, because enveloped viruses are better suited for survival inside the human body, inside the animal host, because they're disguised with a lipid envelope. But outside of the human or animal, these enveloped viruses are not very durable. They don't survive as long because the lipid envelope on their surface, which is outside of the capsid structure, is made of fairly fragile, easily disrupted lipids. So these fragile lipids can be disrupted by most antiseptics and disinfectants. Even just when the virus dries out outside the human body, the envelope may be denatured just enough to no longer be viable in terms of the virus being neutralized. And gram-positive bacteria are just above this, and we can say that if they have any resistance at all on this spectrum of resistance, it would be their thick peptidoglycan that they have. Here's a table listing various antimicrobial chemicals, and these chemicals have very good activity against most bacteria and other microorganisms, but very low effectiveness or activity against endospores and mycobacteria. It's not here for you to memorize. It's just here as a reference and really as a way to provide some perspective at just how incredibly resistant endospores and mycobacteria can be as forms of bacterial life that are not easily controlled by most chemicals that we rely on out there to control microorganisms. This section of the lecture will focus on antibiotics, and here are a few key definitions that we should keep in mind while learning about this part of the chapter. Chemotherapy, first of all, is a type of treatment that we often think of for cancer, but technically antibiotics also qualify as a type of chemotherapy, but for infection. And antibiotics they typically come from organisms. Nature basically invented antibiotics, and then we discovered that there are various bacteria and or fungi that can produce substances that will work to limit the growth of or kill other microorganisms. And the term selective toxicity is referring to a property that an antibiotic or whichever antimicrobial agent that would be taken by the patient would be less harmful to the patient or non-toxic and very harmful or limiting to the infectious microorganism that you're attempting to treat. And this can be measured in pharmacology class as something called the therapeutic index. And the therapeutic index is sometimes abbreviated TI. And it has a mathematical calculation, but we won't be calculating that in this class. Yet, I still wanted to take time to mention that there is this measurement, therapeutic index. And the way that it works is intuitive in terms of its score. The scale is from 1 to 10. And the higher the value, the better. So the higher the value, the better it is at being selectively toxic. Here's a table listing various antibiotics and the sources that they come from as far as living organisms that produce these antibiotics that are in the right-hand column. And some of them would be immediately familiar, like penicillin comes from Penicillium, the fungus. And if there was one organism that comes up the most for being possibly the most valuable, it would probably be Streptomyces for making the most different number of antibiotics that are out there. And there's also something else that we could talk about on this, which is the subject of antibiotic cocktails. So sometimes more than one antibiotic can be given in a solution or in a treatment. And on this table, we could look at the antibiotic cocktail known as Neosporin. So bacitracin and polymyxin and neomycin together are found in the antibiotic ointment that a lot of people have heard of called Neosporin. And this is used in topical applications to prevent infection in minor cuts and scrapes. And you can't take these drugs internally because we'll see that some drugs are not tolerated well by the human body internally. The kidneys or the liver can suffer depending on the antibiotics, the way that antibiotic is processed by the body. But Neosporin is highly valuable for its ability to prevent skin infection and is applied topically. Is very effective. The spectrum of bacteria that antibiotics can treat can be defined as either broad spectrum or narrow spectrum, and so we should define these terms. A broad spectrum antibiotic is defined as one that can kill or inhibit a wide variety of bacteria. And a common way of defining that would be an antibiotic that could kill or treat both gram-positive and gram-negative bacteria. And this class of antibiotics is extremely useful for unknown bacterial infections or bacterial infections that may be tough to treat, and so a broad-spectrum drug may be appropriate. On the other hand, narrow-spectrum antibiotics can be very useful as well when the infection is known and there may be a well-targeted application in the category of a narrow-spectrum antibiotic, which is defined as an antibiotic that kills or inhibits only specific types of bacteria. And these types of drugs are important because they can be used to treat bacterial infections without disrupting as many normal flora or beneficial bacteria. So these narrow spectrum antibiotics, one example or a way of defining them by example could be antibiotics that only treat gram-positive bacteria. And they'll still disrupt some normal flora, but maybe not as much as but definitely not as much as a broad spectrum antibiotic by comparison. And while we're on this subject, we should define the term superinfection. A superinfection may occur after the administration of broad spectrum antibiotics, most commonly where the drug is used to try to eliminate the infection, but not all of the microorganisms may be killed, or a large number of normal flora are killed, and it creates an opportunity for oftentimes drug-resistant bacteria to pop up. So an example of this, a very common example, would be methicillin-resistant Staph aureus, known as MRSA. And methicillin-resistant Staph aureus now represents roughly 1 in 4 hospital-acquired or hospital-associated staph infections. And these so-called superbugs, as they're referred to sometimes, can pop up as a result of multiple treatments with antibiotics of sorts. And they're also placed in the category with other superbugs that we've seen in recent years, such as CRE. CRE stands for carbapenem-resistant Enterobacteriaceae. And one such organism in this group is Klebsiella. And this is a gut bacterium that has been exposed to many different antibiotics, including a valuable antibiotic called carbapenem. And when that drug no longer works, there have been some deadly infections that Klebsiella has gotten out of the gut and into the lungs. Its species name pneumoniae shows us that it can infect the lungs. And when carbapenem doesn't work, oftentimes there's no drug that works. And so there have been some deadly infections in that category. Also in the category of superinfections, there are the C. diff bacteria, and they also live in the gut, along with organisms like Klebsiella. But C. diff make endospores, and these Clostridium bacteria oftentimes will explode in their numbers when normal flora bacteria that do not make endospores are killed by broad-spectrum antibiotics. And then when the broad-spectrum antibiotic is withdrawn, the C. diff endospores will germinate and overpopulate the intestines and cause sometimes severe or even deadly infections that can get into the blood. and they're not always responsive to antibiotics. And also in this category are yeast infections, but not the common yeast infection that may come and go and may only be present in the vaginal area of women, but sometimes yeast infections can get into people's catheters and sometimes into the bloodstream. And these types of yeast infections can be rapidly fatal, and unfortunately, the antifungal drugs that are administered may not be effective in time or at all. And so they are a type of superinfection as well. And lastly on this list, I thought I would take time to mention vancomycin-resistant enterococcus. And these are gut bacteria as well, enterococci. And what they do is they're exposed to a lot of antibiotics that people take. And the antibiotics can be in the gut as they're ingested. And the enterococci have developed resistance to a very powerful drug called vancomycin. And when vancomycin doesn't work, maybe no drug will. So for reference here, I've written in the genus of each organism associated with these acronyms. Here's a table purely for reference. You don't have to memorize this, but it shows some broad spectrum drugs when compared to more narrow spectrum drugs. And so tetracycline would be an example of a broad spectrum antibiotic, as it can kill or treat both gram-positive and gram-negative infections. And then penicillin would be an example of a narrow. And there are many examples of both broad and narrow spectrum antibiotics, but that just shows you a couple of easy to see examples here on this table. Here are the definitions of bactericidal and bacteriostatic, and keep this in mind as we go over the various antibiotics in the rest of this lecture. This is a highly valuable, detailed figure taken from our textbook, and I'm going to recommend that, unlike many other figures that are used purely for reference, this is one figure that you should make some effort to memorize. So we'll be making a table together during our Zoom class meeting, and we'll dissect this. We'll go through each one of the various actions of antibiotic, antimicrobial drugs. And so on this figure, we can see that there are 5 different types of action that antimicrobial drugs can exert, or possibilities for how they work. And so we'll be looking at each one of these in detail as we go over the next few slides. The first mode that we can describe further from the previous figure shown is inhibition of cell wall synthesis. And so some antibiotics inhibit the cell wall, and this is a great target inside of the human because we don't have cell walls. And so most bacteria, the vast majority, have cell walls, and those structures, when they're inhibited, will cause the bacteria to die as they're growing and multiplying. And there are some bacteria that have enzymes that can neutralize this. So antibiotic resistance has popped up in the form of an enzyme that is called penicillinase. And this isn't too surprising in the sense that penicillin was the first antibiotic discovered by humans and put into use. And the enzyme penicillinase has been found to be present in some resistant organisms that have figured out a way to neutralize the penicillin that would normally go in and inhibit peptidoglycan formation. And so penicillin is an example of a cell wall synthesis inhibitor, and then there are the various derivatives of penicillin. And so we'll see in the coming slides here about some examples. Taking a closer look at penicillin and some of its derivatives, we can see that there's penicillin G. This is the original penicillin that initially required an injectable application. So whether it be through IV or through a needle, hypodermic needle, we can see that it has this common nucleus as its molecular structure shows, where there is something called a beta-lactam ring. And the beta-lactam ring is the area that makes penicillin effective at inhibiting the cross-linking between strands of peptidoglycan. The strands, if you recall from chapter 4, are made of NAG and NAM, and acetylglucosamine and acetylmuramic acid. And those strands of NAG and NAM are hooked together by what are called tetrapeptide chains. And the tetrapeptide chains are not effectively able to be linked because of this beta-lactam ring. And the side chain that's over here is how penicillin varies in its different forms. So penicillin V, which was developed later, is a common form of penicillin that can be taken orally. And so these are what are called natural forms of penicillin that are relatively unmodified or original to what was first discovered and developed. And interestingly, the only difference between penicillin G and penicillin V is just one single oxygen atom that we see here in its side chain. And yet, this makes the difference between being able to take it by mouth or that it be required to be administered as an injectable. Semisynthetic penicillins are modified quite extensively in their side chains. They still contain the same beta-lactam ring in their nucleus, and then we can see in oxacillin, by example, this very unusual side chain structure that allows it to be resistant to penicillinase, yet still considered to be narrow in its spectrum of activity against only gram-positive organisms. Oxacillin, in particular, has been one of the most useful drugs in treating MRSA-type infections. Unfortunately, when oxacillin does not work, it identifies that the MRSA infections must be treated by something additionally, maybe vancomycin, for example, or some other type of alternative antibiotic. Ampicillin has a simpler side chain, but ampicillin is known to be an extended spectrum type of penicillin that can treat many types of gram-negative infection in addition to treating gram-positive infections. When we look at penicillinase specifically as an enzyme that can break down penicillin antibiotic, we can think of the penicillin as a substrate. By which the penicillinase enzyme will act. Now, penicillinase, interestingly, can act as an endoenzyme when it is found in periplasmic space, which is the area right above the cell membrane and in between the cell membrane and the cell wall, or it can actually be secreted. So it depends on the species and how the species of organism that produces the penicillinase is making use of it. If it's an exoenzyme, that means that it's secreted outside of the organism that produces it, and it may seek then to neutralize the antibiotic before it enters and becomes dangerous to the organism. The periplasmic space would be considered inside the organism, and so that would be an internal endoenzyme mode of action. Penicilloic acid is considered to be the end product And this forms when the beta-lactam ring is broken. And that is the key to penicillinase's activity, is that it breaks the beta-lactam ring and it then neutralizes the antibiotic. So I'll label that down here, broken beta-lactam ring. It should also be noted that beta-lactam antibiotics are not limited to just penicillins and its derivatives. It turns out that cephalosporin drugs, which are produced by a different fungus, not the penicillium fungus that produces penicillin, but cephalosporium. And cephalosporium happens to have the same beta-lactam ring in its mode of action as a cell wall synthesis inhibitor. But the nucleus that contains that beta-lactam ring has a different molecular structure. Other cell wall synthesis inhibitors include what are called polypeptide antibiotics. And the two polypeptide antibiotics listed here are bacitracin and vancomycin. Bacitracin was mentioned earlier as an ingredient in Neosporin, the triple antibiotic ointment, and it is limited to topical applications only. It is not tolerated well by the human body internally, and so it is only FDA approved for administration topically, and it is excellent at treating gram-positive bacteria. It does not show effectiveness against gram-negative bacteria as far as I understand. So we label it as being good against gram-positive, especially skin bacteria. So think organisms like staph. Vancomycin is a type of antibiotic that more specifically is referred to as a glycopeptide antibiotic. Glyco refers to sugar, but it is also still peptide based. So it's part sugar, part amino acid based. And it's been found to be extremely useful against treating drug-resistant staph infections. So MRSA, methicillin-resistant Staph aureus, and I wish I could tell you that vancomycin worked every time under all circumstances, but it doesn't. And in these cases, which are oftentimes difficult to almost impossible to treat, people may develop what's called VISA. VISA stands for vancomycin resistant. Vancomycin intermediate Staph aureus. And the issue here is that vancomycin is a drug that is not well tolerated by the human body. So the liver really can't tolerate a drug dose that's higher than normal. So by the time we get to vancomycin intermediate levels of resistant in Staph aureus, then this is really the end of the line, because elevated doses just aren't going to be possible in someone who you're treating in this case. There's also vancomycin resistant enterococcus mentioned earlier, called VRE, and it's responsible for superinfections, drug-resistant infections. And so I wanted to make mention of these particular organisms with respect to vancomycin, which is a cell wall synthesis inhibitor, as we know. Other cell wall synthesis inhibitors include the drugs to treat TB and leprosy. These are antimycobacterial antibiotics, and isoniazid is considered the first line most important of those. It's a go-to drug for treating TB pretty much always, and this is because isoniazid inhibits mycolic acid synthesis. And if Mycobacterium does not have the ability to make its mycolic acid, then its cell wall structures will be inhibited during their formation. I just spelled the wrong thing here. It inhibits mycolic acid synthesis. And isoniazid is a drug that is typically given for long periods of time. And this is because of the slow growing characteristic of TB and mycobacteria type bacteria. They grow and multiply about once a day, which is an extremely slow rate. And so a long administration of a minimum of 2 months up to 6 months on average is the standard for treating these bacteria to hopefully eliminate them before they would cause infection in people who test positive for their TB skin test or their blood work or chest x-rays. And so in addition to these drugs, or to this drug given for TB and leprosy, is the ethambutol as a drug that is only reserved really for when people need it. And so if there are drug-resistant strains of TB, which there are, if they do test positive for drug resistance, ethambutol is given because what it does is it inhibits mycolic acid deposition. So incorporation into the cell wall is inhibited. So even if mycolic acid somehow manages to get made by the mycobacteria, then the ethambutol will inhibit the incorporation of any mycolic acid. And that'll still then serve to inhibit cell wall synthesis. So because it's hitting cell wall synthesis at a different angle, this is referred to as a synergistic kind of characteristic, working on the same pathway but from a different angle. One of the drawbacks, though, is that although it is synergistic with isoniazid, it's reported to have a lot of side effects. So depending on the person and their ability to tolerate this drug, these side effects may be very difficult to manage. But if you're talking about getting rid of a life-threatening mycobacterium infection or a drug-resistant mycobacterium, then ethambutol can still be very valuable to life-saving. A second class of antibiotics are the protein synthesis inhibitors. And these protein synthesis inhibitors work on the ribosome. So whether it be large subunit of the 50S ribosome of bacteria, or the small 30S subunit, or maybe just looking at the ribosome altogether as a 70S bacterial ribosome, the protein synthesis inhibitors will bind to the ribosome in some way, shape, or form and inhibit the bacterium's ability to make proteins. And so this is used for reference. We do not have to memorize that chloramphenicol binds the 50S large portion, or that streptomycin binds the 30S small subunit, or even, interestingly, that tetracyclines block the messenger RNA from sliding through the whole ribosome, functionally speaking. We can take a look at this and understand that protein synthesis inhibitors can have various modes, various attack points within their mode of action as protein synthesis inhibitors. The remaining 3 modes out of the 5 modes of antimicrobial inhibition will be covered during our Zoom meeting when we discuss the 5 modes altogether and make a summary table. For now, let's move on and look at the desirable characteristics of various antimicrobials. So a few of these that should be noted are the fact that side effects are a very important characteristic that must be addressed and hopefully avoided when possible by giving antibiotics that make the most sense for that individual patient. And some antibiotics, some drugs are well known to have a variety of side effects. Other drugs we know are well tolerated by most people. But each individual should be taken into account by the doctors and the nurses to figure out how to give drugs that would have the highest therapeutic index when possible. So a high TI is desirable, of course. Okay. Another aspect that's desirable with respect to antibiotics would be a fast-acting nature. So we would hope that when a drug is given, that it will have a swift and high level of therapeutic effect, and maybe also long-acting. It's desirable to have a drug that has the ability to stay in the system for a longer period of time so that fewer pills would have to be taken. A drug that requires a patient to take the drug 4 times a day would be tougher to comply with than a drug that maybe is only taken once a day. Also, how is that drug administered? The means of administration is an important aspect of a given drug. By mouth or sending a patient home with pills is much easier and less costly to administer than a drug that may only be given by injectable parenteral administration, which would be like a needle or by IV. Another characteristic that we can think about is good diffusion. So how well does the drug make its way into the various tissues of the body or the bloodstream? Does it achieve a good blood concentration or tissue solubility? Another factor to consider would be whether the drug has a cidal effect, where it kills the microorganisms that it comes into contact with, versus a static drug. And it's good to have options because some situations may require a cidal or a static drug as an option, one versus the other, and it's very specific to what's being treated. And narrow spectrum drugs are always nice to have. So less disruption to normal flora. This is a good thing when it's possible. And finally, drugs can be very expensive, and it's quite nice and desirable if a drug has become more affordable. So drugs that are inexpensive, maybe the generic is available or there isn't the patent may expire on a drug, and then that drug becomes more widely available at less cost. The next few slides summarize the laboratory techniques that are employed in lab, but I thought I would still mention a little bit in lecture, like the Kirby-Bauer method, or as it's sometimes called, the disc diffusion method. And this is a strategy in which an oversized Petri plate, typically a large enough plate to fit a wide variety of antibiotic discs, can then be tested for their effectiveness against a given microorganism. And when we look at this particular antibiotic disc diffusion experiment, we can see a fine line, a line between where inhibition is achieved and then no longer achieved. And this is what's called the MIC. The MIC is an abbreviation for the minimum inhibitory concentration. This is the concentration where the drug no longer has an inhibitory effect. So if we look at a given drug, like the one on this disc here, that particular area has the highest concentration of the drug, right where the disc is. That's where the bomb dropped, so to speak. And as the drug diffuses outward from the center of the disc and then further and further out, there will be a decreasing, dropping percentage or concentration of the drug until the concentration of the drug finally drops to a minimum that no longer inhibits bacterial growth. And this minimum inhibitory concentration is, in concept, just a line that we see here between growth and no growth. But there is a number, an actual micrograms per mil or milligrams of drug that could be given to someone in which the MIC is achieved. And we would like to know what that number is. So this plate shows us visually where it exists, but we don't have the number, we don't have that measurement. So other laboratory techniques can be taken to measure the MIC. The broth dilution test is a good old-fashioned method that makes this possible. You can take what are called well plates, and they're little plates with small volumes of liquid that can be held, where you could take a drug and start with a high concentration and then half that concentration and half it again and half it again and half it again, by example. So here's a drug like kanamycin, for instance, and kanamycin may start out at say 200 milligrams per mil here, or micrograms, and then it could drop to 100 and then 50 and then 25 and then maybe 12.5 here. And so the minimum inhibitory concentration would be the amount of drug that still inhibits the growth, and below that there will not be inhibition. And the broth dilution test can become technical, like here. This particular setup for sulfamethoxazole, a sulfa drug, describes that there is what's called the trailing endpoint. And it says this is usually read where there is an estimated 80% reduction in growth. So a drug does not have to be all or nothing, but the point at which the growth shrinks down to 80% less would be considered still extremely effective, and may be effective enough for the drug to actually be read as an MIC in that case. So this is the broth dilution test, and it requires a lot of pipetting and calculating the various concentrations of the drugs and mixing up all of these different solutions at different dilutions. Now, an easier technique, that the E-test, which is in reference to an epsilometer, is a more clever, efficient way to determine the MIC. It revolves around using a plastic strip which has various levels of antibiotic of a given drug in it, as opposed to using a disc. So it is not a disc diffusion method, but a strip diffusion method. Pictured here, we can see the E-test in its application. So we see the elliptical shape around the antibiotic strip, and so it has this unusual shape. And this is a product of the varying concentrations, starting with the highest concentration up here at 256. This strip says TM on it, which is in reference to an antibiotic. I believe it's telithromycin. And this antibiotic starts out at 256 micrograms here, and then drops to 192, and then 128, and on downward until there's less than a microgram present in this part of the strip. And this is labeled in this figure as to the MIC. So the MIC is the minimum inhibitory concentration, which happens to be 0.064 micrograms of telithromycin. And so this is one of the most efficient, best ways to determine an MIC, and it is called the E-test. There are 4 mechanisms by which bacteria can physically resist antibiotics. These 4 mechanisms include physically blocking the antibiotic from entering, and this depends on the species and how it achieves this. We know that there are certain species of bacteria that can form slime or capsules and make biofilms. So that would be one way that bacteria physically resist the entrance of an antibiotic into their environment. And we know that other bacteria may make endospores. That would be another mechanism. Even just the gram-negative outer membrane may be a mechanism for blocking certain antibiotics. There are many antibiotics that will not pass through the gram-negative outer membrane. But whatever the case, these physical structures will block antibiotics from getting in. And a second mechanism that bacteria can use to physically resist is that even if the antibiotic gets in, there could be an enzyme that inactivates the antibiotic. And one of the best examples that we know about would be the penicillinase enzyme. So penicillinase can break down penicillin, and then penicillin, there are many derivatives of penicillin, and so collectively we can talk about various penicillinases as beta-lactamases, because the way that they work is they break down the beta-lactam ring within the penicillin-based antibiotic nucleuses and in their molecular structure. And then number 3, there is the alteration of a physical structure or some sort of chemical structure within the bacteria. And this requires that the bacteria mutate in some way. So mutation will bring about a physical change in the bacterium's structure, either molecularly or in its anatomical makeup. And lastly, if an antibiotic gets in, some bacteria have engineered a way to kick the antibiotic right back out. So as soon as it comes in, there are sometimes what are called pumps, efflux pumps, which can obtain the antibiotic after it goes in, grab it, and kick it right back out. So these are 4 ways that we know about that bacteria can physically resist antibiotics as they either are attempting to enter, or if they enter, or even if they're then kicked right back out. Another important aspect of antibiotic resistance is how bacteria are able to acquire the forms of physical resistance that were listed in the last slide. And it's always going to come in some sort of change to the DNA itself. And so there are 4 genetic mechanisms that we should take note of. So I'll write up here, mechanisms of genetic resistance. And the first way that bacteria could mutate genetically could be just simply through random mutation. Random mutation is sometimes referred to as spontaneous because bacteria have a frequency of mutation that is fairly constant even without any kind of unusual event or even a planned event. This is just an unplanned natural phenomenon known as random or spontaneous mutation. However, other forms of mutation may be more purposeful, such as conjugation. Conjugation, we know from chapter 4 in this class, involves the sex pilus. So you have bacteria that will either pull up alongside one another and copy DNA from donor to recipient through a sex pilus, or perhaps a mating bridge. So if there's no sex pilus, we know that gram-positives, which don't form pili, could form a mating bridge. I started writing that wrong. We have a mating bridge. And another way that genetic resistance can be acquired is through a process called transformation. Transformation is rather mysterious. At least it was in concept for a long time, but we now know that bacteria have been discovered to have the capability to uptake naked DNA from their environment. So imagine a dead donor bacterium that could have been drug resistant, but maybe it just died of old age. And this bacterium spills out its DNA after it lyses, and that DNA may contain antibiotic resistance genes that can be picked up by a recipient that undergoes what's called transformation. And finally, there is one last possibility where resistance genes are transferred through a process called transduction. Transduction is facilitated by what are called phage viruses. Phage viruses are viruses that can infect bacteria, and sometimes they act as a genetic vector, which means they can pick up DNA from a current host and bring it to a future host. And so then antibiotic resistance genes are sometimes transferred from a previous bacterial viral infection with the phage that then picks it up and brings it to a new bacterium that becomes a recipient, not just of viral DNA, but of bacterial resistance, antibiotic resistance genes. Here is a fairly simple figure that shows the evolution of antibiotic resistant populations through natural selection. At the top, what we see are a population of bacteria that could be causing an infection. And within that population, the majority of bacteria would start out as S labeled as sensitive to a given antibiotic. However, a small number, maybe a few members of that population could actually be R, which stands for resistant in this case. And if there are one or more resistant members of the population, then they may survive the administration of a given antibiotic. So initially, at this first phase, we can label this antibiotic administration. So an antibiotic is given, and the normally expected outcome would be that the infection would be killed off. But if that does not happen and antibiotic resistance builds up, then we'll see the sequence of events that happen next. So we can see in the second phase that the sensitive bacteria do die. And they're labeled as dying because their cell walls are breaking apart here and lysing. But there's one resistor left over, and the one that's labeled R that doesn't have a dashed or dotted line around its cell wall may survive, and then if it continues to grow because the antibiotic does not have an effect on that organism, then eventually there can be a superinfection where only resistant bacteria labeled R are now causing the infection. And so temporarily the patient may feel that they're getting better or that there were improvement in symptoms, but then the remaining resistant bacteria end up eventually roaring back as what is sometimes described as a superinfection. And then a new antibiotic will have to be used to try to get rid of these resisting bacteria. It's important to note that the bacteria are what are evolving resistance when antibiotics are given. It's not about the patient physically resisting the antibiotics. Sometimes it's said that the patient is resisting the antibiotics, but in fact, it's the bacteria inside of the patient that are doing the resisting. This is an example of how an antibiotic-resistant population could manifest over time. So what we see in this graph is a measurement of bacteria in the person's blood on day 1. So to begin with, it's very high, and this person has an infection. So at such high levels, doctors decided to give an antibiotic, and as they administered the antibiotic, over the next few days, the numbers of bacteria in the blood started to drop dramatically. So after 4 days, you can imagine that this person's symptoms and feeling of well-being started to improve, but then something dramatically different happens. Transitioning between day 4 and day 5, it says on this other measurement of the y-axis that antibiotic-resistant bacteria started to increase in the numbers in the blood in terms of milligrams per mil of drug that was necessary to be given. The bacteria started to rise despite ever-increasing levels of antibiotic. So antibiotic resistance went way up, and the numbers of bacteria began to climb. When they were once dropping, they started to rebound, and this is showing visually the presence of a superinfection and how essentially the drug studied in this figure would no longer be effective. and so a different antibiotic would have to be administered. Lastly, we should list several ways in which we could stop the development of antibiotic-resistant bacteria in a given environment or in a patient or just in general. How can we stop antibiotic resistance from developing? One major piece of advice I would say we should all follow is take your complete prescription of an antibiotic. If a doctor gives you a certain number of pills, then that's how many pills are necessary as determined through clinical trials. The complete prescription will ensure that you apply the pressure, whatever it is that you're trying to do with that drug to kill the bacteria, that you want to take that drug on time, every time, for the whole prescription. People often make mistakes in this regard where they'll take the drug until they feel better, or just a little bit longer until the symptoms are gone, and then they do not take any more of the drug. They might decide to take the drug and put it in the medicine cabinet. So the remaining pills will sit there, and they figure they might use them at a time when maybe they would need them again someday. But this isn't good, because then you have half a prescription taken and the other half still not taken, and neither time will a complete drug administration be given. And this can lead sometimes to resistors not being completely knocked out, and superinfections sometimes do result, and then the antibiotics have no use at all. Secondly, it's advised that we discard leftovers. So discarding leftover antibiotics is related to the same topic, that if you don't have a complete prescription, then it may not be advisable to take only part of a prescription. Again, the risk is resistant bacteria developing because not enough of the drug was given for long enough. Now, on the other end of the spectrum, it's also important that we avoid overprescribing antibiotics. Now, this is the doctor's responsibility. However, overprescribing antibiotics has led to increased levels of antibiotic-resistant bacteria, globally speaking, in areas of the world where antibiotics are given too often, or just indiscriminately, or prophylactically. And it's not only a problem in humans, but also in livestock. Antibiotics have been given in high volumes in animal feed over the years. And this can be a big problem, because although it may avoid bacterial infection that can set back farmers when they're raising livestock, but then it can bring about antibiotic resistance, because these drugs are given at times when they weren't even necessary. And this antibiotic resistance has been shown to pop up then in its consumers. Unfortunately, antibiotic-resistant bacteria may be in or on those animal products, or those antibiotics may be in the meat or the livestock that we're consuming. And that's not good either, because then we have excessive exposure to antibiotics. Vaccines are another big issue these days, and in the past and the future. We need vaccines in order to positively affect how many infections happen amongst us as humans. And when you're up on your vaccines, it's likely that you'll have fewer infections, at least for the infections that vaccines can prevent. And so that means fewer antibiotics administered. Additionally, just some of the common sensibilities of having a proper diet where you don't have nutritional deficiencies that can lead to getting sick more often, and exercise. I think it can't be stated enough that physical fitness is not only making a physically strong body that you can visibly see, but a person who's fit also has a better immune strength. And sleep is so important in preventing stress on the body. People who are low on sleep and who are not resting properly will have reduced immunity, and this will result in more antibiotics given as well to people who get sick more often. And finally, we should really only take antibiotics when the doctor deems that it's necessary. So we can say we should take antibiotics only when doctors prescribe them or recommend that we should. It's been observed that outside the United States, where antibiotics can be bought at the pharmacy without a doctor's prescription, that the levels of antibiotic resistance have grown more quickly, where people take antibiotics more often than they need to because they're not really using a doctor's judgment. They're just using individual judgment, which may not be as prudent or as conservative as what doctors would recommend.